Transduction. I miss IR a lot. This is for Bio 515, Human Physiology 1, Professor Dr. Barrera. Our objectives. What is phototransduction? Phototransduction in the peripheral nervous system. We'll discuss photoreceptors, information from recent studies, which will include rod receptor cells, photoreceptor and bipolar cells, materials and methods, data, results, and then we'll have our conclusion. Phototransduction is the energy from light being converted into electrical signals in the retina. This allows an image to be seen through the pupil. While phototransduction is a factor for images being seen, also are rhodopsin, the visual pigment PDE, called phosphodiesterase. Altogether, they help the visual images get processed in the body inside rod cells. Phototransduction within our peripheral nervous system happens in the rod and cone cells within the retina called photoreceptors. These photoreceptors are neurons that help images be seen. Light is then reflected off the object, and the rod and cone cells convert that light into electrical signals. From here, these signals are then processed through our brain, and it allows us to process the image. Photoreceptors are activated by light being reflected, and the cell starts to depolarize. Within the shining light are the rod or cone cells. Now, once the cell membrane depolarizes, this eventually will stimulate an action potential, and the neurotransmitter releases the neuron it contacts. Within the retina, the photoreceptors wait and show action potentials, and then light activation will cause a graded change in membrane potential. As this is being transmitted, the retina is mediated by graded potentials. Let's look at this figure. Here we have an anticellular recording from a single cone stimulated with different amounts of light. We have membrane potential from -65 to -40. Our time is going in from 0 up to 600. You have our light flash. This data shown is from the retina of a turtle. It shows relatively long time course of response. Each trace represents the response of a brief flash that changes with intensity. At the highest light levels, the response is amplified and saturated at -65 MV. That's so we get our most intense flash response. The hyperpolarizing response is characteristic of photoreceptors from organisms that are vertebrates. In recent studies, rod cells are located inside the retina and have learned to adapt to dysfunction. The changes that happen in photoreceptors cell membranes can respond to signaling being disrupted, specifically from diseases like night blindness. Rod receptor cells, main type of cell located in the retina. Vision itself begins in the RS, or a rod outer segment. Rhodopsin is produced in the retina, which is a protein, and it consists of a protein, which is opsin, connected to the 11 cis retinal. Here, it locks itself into an inactive state, and once being activated, the like protein is signaled, and this is phototransduction. Let's take a look at figure 1. Here we have photoreceptor and bipolar cells. Photoreceptor cells must adapt to changes in the lighting environment or any dysfunctional state. Signals from rod receptor cells are passed on to rod bipolar cells, and they are located in the outer plexiform layer of the retina. Here is a figure of photoreceptor and bipolar cells. Here we can see that the rod photoreceptor cell has an outer segment containing stacks of discs that are encased by a plasma membrane. Rhodopsin is packed into the membrane's disc. This is within these discs, which we have in red. It initiates phototransduction upon photon capture. The rod photoreceptor cells make synaptic connections to the rod bipolar cells, which are located at the bottom. Changes in the glutamate release, which is within the bipolar cell, are detected, which are in green, and the synapses of the rod bipolar cells. In this study, genetically modified mice were used and rod outer segment disc membranes were isolated. Procedures were carried out under dim red light conditions, and 8 to 15 dark mice were examined for CSNB, which means congenital stationary night blindness. Let's take a look at our table. Here on our table, we have our mouse models, which consist of 4 on this particular model. And there are 4 diseases. Those diseases are ARCSNB, which is autosomal recessive CSNB, and AD is autosomal dominant, and LCA is for Leber congenital amaurosis. Our models are the GNAT1, the ROG90D and 90D again, the RPE65, and the GRM6. Okay. And again, we have the diseases. The dysfunction from these diseases or the elimination of transduction prevents phototransduction. Photo receptor cell signaling is eliminated. G90 mutation in rhodopsin causes receptor to be constitutively active. Photoreceptor cell is desensitized. Elimination of RPE65 prevents 11 cis retinal regeneration. Rhodopsin is present only in the apoprotein opsin form, which is constitutively active. Photoreceptor cell is desensitized. Elimination of mGlu-R6 prevents postsynaptic signaling in bipolar cells. And last, signaling from photoreceptor cells to bipolar cells is eliminated. Here are no adaptations for of mice expressing G90D. ROS disc membranes were prepared for row G90 and G90D mice. These mice were housed under normal cyclic light conditions. The ROS disc membranes were imaged by AFM. AFM is autonomic force microscopy. And here you can see that they're exhibited a characteristic rim and lamellar region containing nanodomains of oligomeric rhodopsin. and it observed the nanodomain organization of rhodopsin is not the artifact of AFM procedures and similar arrangement when observed by cryo-electron microscopy. Let's take a real look at our figures. Again, our figures are sampling of AFM images. Here you can see that from A to C, you have row G90D, and D to F. D to F is row G90D, and D to F is RPE65. The G through I mice, down for RPE65, are housed under cyclic light conditions. And then the rim for region 1, which is A. And then there's a lamellar region that contains the rhodopsin nanodomains. Is 2, is highlighted here. RS disc properties for wild type and row G90D, as well as RPE65. These mouse, again, were housed under cyclic light conditions. And the mice were housed for 10 days in constant darkness. This data consists of looking at the following: the disk size, nanodomain number, rhodopsin number, nanodomain density, and rhodopsin density. The mice were housed in either constant light conditions and constant dark conditions, exhibiting opposing adaptations. Under constant dark conditions, when light was absent, photoreceptor cells increased rhodopsin density, as you can see here, in the RS disc membranes. To determine if changing the environmental lighting condition could promote adaptation in photoreception cells, the row G90D and RPE65 mice were housed in constant darkness again for 10 days, similar to the mice housed under cyclic light conditions. There were no significant differences observed in RS disc properties. Thus, the photoreceptor cells expressing either constitutively active G90 rhodopsin or absent or incapable of adapting to long-term environmental lighting challenges. So if we take a look at each one of our charts gives us the models that we're using, and a wild type of mice is being used as a reference in all of these charts. So statistically, there are no significant differences with these group of models. As we continue to move through the data, there could be a possible change. Adaptations of mice expressing GRM6. Again, these images are taken for AFM images, which is autonomic force microscopy. Although impairment of phototransduction was previously shown to promote adaptations in the RS disc, it's unclear whether or not signaling to the bipolar cells is necessary for these adaptations in photoreceptor cells. These RS disc membranes, such as GRM6, the mice were housed under cyclic light conditions and were examined by the AFM imaging. Like other mice being examined, RS discs were properly formed with rhodopsin forming nanodomains. From our figure here, we can see that, again, they're being housed in darkness for 10 days. And from looking at in this figure, D through F, shows us no further changes when both phototransduction and signaling to bipolar cells are impaired. We're still not quite seeing a change or an adaptation, and they're still in dark conditions for 10 days. Here, RS-DIS properties consist of GRM6, GNAT1, and GRM6, again, housed under cyclic light conditions, again, housed for 10 days in complete darkness. Although the GRM6 mice exhibit a large mean nanodomain size, an individual RS-DIS, the distribution of nanodomain size is skewed, and it could be a difference in mean nanodomain size, and it may only reflect subtle differences in the sizes of rhodopsin nanodomains that have been formed. Here you can take a look at our chart, and we can now see some differences and some adaptations that allow, because of the environment in which our nanodomain size increased for GRM6. There was no change in the number for GRM6. And when we take a look at our GRM6 for nanodomain density, it increased. It increased and it exhibited different adaptations compared to that of the GNAT1. Whether it's the regular mouse models, the wild type mice housed in constant darkness from birth. This difference indicates that the lighting environment or signaling capacity of photoreceptor cells at birth or during early rearing of mice can impact adaptations in photoreceptor cells. In the mice. Final results. Organisms spend their lives adapting. It is needed for survival. Increased rhodopsin nanodomain size has subtle changes. It is dependent on complete darkness or under cyclic light conditions. Mice have different types of CSNB that can result in different adaptations to their environment. And when we take a look at our summary of the changes from looking at row G90D, GNAT1, and GRMB, there's no change observed with our row G90D. Our GNAT1 and GRM saw an increase of the number of nanodomains as well as rhodopsin, nanodomain density, rhodopsin density, nanodomain size, and rhodopsin density. In conclusion, phototransduction plays a vital role in how we can see images. The specialized photoreceptor cells are called rod and cone cells. Phototransduction happens in the retina and starts the process of initiating by seeing the light being reflected and converts it into an electrical signal that is then interpreted by the brain. Certain adaptations from our environment, such as light, can determine how organisms can adapt their vision to their new surroundings. Thank you.